Welcome. Ooh, that's up. Yeah, you can go. All right, welcome. We're going to go ahead and get started. Okay, I'm Kara Foshay. I am Senior Director of Genetic Therapies at the CF Foundation in Bethesda. And this is my colleague, Hillary Valley, who's Mission Ventures Investment Principal at the Foundation. We just got a suggestion that we should start with sharing what we do at the Foundation. And so we are part of the same science and innovation team at the Foundation that looks to identify, evaluate, fund, and then develop the therapies that you guys hear about. And so when you think about that spectrum of how that happens, Hillary is on the early side, going out, meeting with new companies, bringing their technologies back, really leading the evaluation of those therapies. And we overlap as we start to sign a deal. I come in and I help those companies to develop the plan that they're going to move forward, making sure that they have the right tools to develop for CF and help to see those companies as they move forward into clinical trials. And so we really are teammates working together to make this stuff happen. Okay. So during this talk, we're going to build on some complex scientific topics. And so although we normally ask you to save your questions until the end, if you have a question about the science that we're talking about on the screen, we're going to encourage you to raise your hand and ask that live. And we're not going to speak at the same time, so one of us will be down walking around with the mic. If you have general questions about your mutations or specific clinical trial, sort of general things, we'll ask you to save those for the end. But we really do want to make sure everybody's following along with the story we're telling and can stay, come along with the science. So if you do have a question about what we're talking about, feel free to raise your hand. Okay. All right, full house. So with that, we will get started. We'll start today with an overview of genetic therapies and how they work, because I think it's important for everyone to have that good biological understanding as we move forward. We're going to give you an update on the current genetic therapy clinical trials that are ongoing. And then we're really going to take a deep dive and try to help you connect the dots to understand how targeting a cell type and delivering a genetic therapy into that cell can kick off a chain reaction that improves the health of the entire lung and can eventually be measured in a clinical trial through a change in FEV1. How do you connect the cells to health? And finally, we're going to give you a little sneak peek into what's coming next for genetic therapies. So even as we watch clinical trials that are currently ongoing, we don't want to wait before we start thinking of what version 2.0 makes. So we're going to wrap up our talk with a focus on targeted delivery, which we see as the next improvement. Okay. So the flow of genetic information is really important for understanding how cystic fibrosis how mutations in the CFTR gene can cause cystic fibrosis. And it's also important for understanding how genetic therapies can benefit all people with CF. Each cell in our body contains the same genetic code. It's our DNA, our genes. And the sequence for each gene is transcribed into an mRNA, or a copy of that gene's really important key information. mRNA is translated into a functional protein. The CFTR gene encodes for the CFTR protein, which functions as a channel to transport chloride ions outside of the cell. Mutations that occur in DNA are copied into mRNA, and they're translated into protein where they may have detrimental effects on protein function. Mutations in CFTR can result in a CFTR channel that does not transport chloride. Cystic fibrosis can be caused by many different mutations that occur across the very large CFTR gene. The color coding along the top of the gene shows which parts of the gene encode different parts of the protein. This is important because different parts of the protein have more essential functions than others. And so mutations that occur in certain regions that are really important for CFTR function, can be more severe. So how can we correct the flow of genetic information when it's disrupted by a mutation? Starting at the top, we could target therapies to correct the mutation containing DNA using gene therapy or gene editing. Gene therapy provides a new copy of the entire non-mutated CFTR gene that sits inside of your cell, and so it could benefit people with any CF-causing mutation. Gene editing actually goes into your cell and corrects a person's own DNA, and these therapies must be engineered to correct specific mutations. Moving down, approaches to correct mutations at the mRNA level include RNA therapy and antisense oligo or ASO treatments. mRNA therapy delivers a non-mutated CFTR mRNA that sits inside your cell and could benefit people with CF that have any CFTR-causing mutation. Antisense oligos work differently. They bind to a mutation in the mRNA and mask it, allowing for protein production. And because of this, they must be designed to target specific mutations. In CF, mutations like F508del allow cells to make an unstable CFTR protein that doesn't fold or function correctly. Mutations like this, where at least some protein is present, can be treated with small molecule CFTR modulators, but it's important to note that modulators are not considered genetic therapies. Now, some mutations in CFTR prevent any protein from being made at all. And these mutations cannot benefit from modulators. There is no protein for a modulator to bind to. And for people with these types of mutations, genetic therapies are the best hope of a transformative therapy to address the underlying cause of CF. In the last slide, I highlighted several types of genetic therapies, and it's important to note that not all genetic therapies act in the same way. Delivering a copy of mRNA or DNA to the cell is not permanent. These therapies will need to be redosed, and depending on the modality, that could vary from days to even years. Gene editing and integrating viral gene therapies are grouped together as permanent treatments because they change a person's own DNA inside the cells. And these changes will last for the life of the cell in which they're made. And if they're made in a stem cell, which divides and repopulates, that change will be present in all new daughter cells that are made. Both permanent and non-permanent treatments have the potential to become transformative therapies for people with CF. And today, we'll focus here on non-permanent delivery of RNA and DNA therapies, which we'll generally refer to as CFTR replacement therapies. Many of these are in clinical trials now. And although it remains a priority area of research, there are no CFTR gene editing approaches in the clinic today, and so we're not going to dig into that topic in today's talk. So what is a genetic therapy, or what does it look like? Almost all genetic therapies have two key components, a cargo and a delivery vehicle. The cargo is comprised of the genetic material, the RNA or the DNA nucleotides that enter the cell to make a CFTR protein or a gene editing protein. The delivery vehicle helps the gene editing machinery to reach the correct cells. It facilitates uptake into those cells and can also serve as a protective outer coat. And it's important to mention that the delivery vehicle isn't just a wrapper. It's not something we just throw on. It's actually an integral part of the therapy and is one of the most complex pieces of developing these genetic therapies. So when we compare gene replacement, RNA and DNA gene replacement therapies, we consider what would these look like as a medicine. We know that they would both restore CFTR function for people that have any CF-causing mutation. They would both target the differentiated cells of the lung epithelium. So those are the cells that line the lungs and are repopulated over time. All of the CFTR replacement therapies that are currently in clinical trials and under development are targeted to be inhaled therapies, which means they would only address CFTR in the lungs at this point in time. Now, they get to be a little bit different when we think about duration. mRNA is naturally a very unstable molecule, and in the body it lasts only hours or maybe a day. And so we know that dosing would have to take place every 1, 2, or 3 days in order to keep a constant source of CFTR present. DNA is a little bit different as it can be engineered more readily and can have different durations. So we see trials dosing DNA therapies anywhere from 1 week to once or yearly. The key challenge for both of these types of therapies is to find an effective dose that is also safe, and we're going to dig into that today. So as these therapies are developed, they move through this clinical trials pipeline, and what I have here are the phases of a traditional clinical trial. Moving a product from the lab where we study how it works in model systems to first-in-human trials, which are by design small, short studies focused on evaluating the safety of increasing doses. And oftentimes, these will contain, may include healthy volunteers. As we move to phase 2, studies get a little bigger, but they're still focused on evaluating safety, usually in a few more people at a dose that we think could be effective. And so they start to include exploratory endpoints to look for biological activity or early signs of efficacy. But it's not until we get to a traditional phase 3 that trials are actually designed and powered, which means they contain enough people to get statistically significant information, to show safety and efficacy. And so it's phase 3 data that are used to support regulatory approval of a therapy. It's also important to note that once a therapy is approved, long-term surveillance continues to monitor safety in people that take that approved drug. And we see that ongoing now for CFTR modulators. Now, I mentioned this is the traditional path. With small populations like we have for genetic diseases often and for CF, and for innovative therapies, we sometimes see less traditional designs where studies flow seamlessly from phase 1 to 2B. And you'll see that in some of the trials I mentioned. With small numbers of participants, early signs of efficacy can be difficult to observe. So imagine, for example, a clinical trial that's in phase 2 that only has 3 people receiving a certain dose. You guys know about CF. Imagine one of those people with CF gets a cold during the clinical trial. That happens, and their lung function decreases. It's really hard with only 3 people, one of whom now has a cold, to determine whether or not we're seeing signs of efficacy. Now, as a patient advocacy organization, the scientists on our team are constantly monitoring that because we want to make sure that only the therapies with the best chance of success move forward. So what do our current clinical trials look like? I think we often hear bits and pieces, but I tried to put it all together into a table, which I realize is really busy, so take a picture, study it later. But this is the most recent public information we have from any of these trials. And you'll see the things I talked about on the last slide. These are all in phase 1, phase 1/2, or phase 2 studies. And I have the number of participants highlighted, and what you'll see is that they're very small numbers, just as I said. And so while they are looking for early signs of efficacy, again, it's really hard to interpret some of the data. And we're going to aim in this talk to help you have the biological understanding to interpret what you see when it comes out. So at the top, we have SPL84, which is SpliceSense's antisense oligo designed to target the 3849 plus 10 KB mutation. And we're particularly excited because SpliceSense has recently reported an increase in FEV1, which is the first improvement in FEV1 we've seen for a genetic therapy in CF. And we expect to see more data coming from SpliceSense later in 2026. In the blue, we have our 3 mRNA lipid nanoparticle trials from Arcturus, Recode, and Vertex. We've heard the most information from Vertex so far, who reported that they unfortunately didn't see a change in FEV1 over 28 days with their middle dose, which was 10 milligrams. This was in 6 patients. Small numbers, hard to see efficacy. In the coming year, they're going to complete their high-dose cohort, which is 15 milligrams, and they're going to begin an expanded safety study that'll last 12 weeks. So we hope to see more data from them toward the end of the year. Now, with Vertex, I think it's worth mentioning that they paused their trial in 2025 due to an adverse event, but they restarted enrollment. And the reason I highlight this is to highlight the role of the DSMB, the Data Safety Monitoring Board. It's something that you should know exists for all clinical trials that are ongoing. An external panel of experts that review safety data as it occurs and make a decision as to whether or not the trial should proceed. In this case, they decided the Vertex trial should proceed, but I thought it was important for you to know that this is something that happens, is present, and is another layer of protection for all the people we know in CF trials, in any clinical trial. At the bottom, we have our gene replacement therapies from 4D Molecular, which has an AAV or adeno-associated viral CFTR replacement therapy, and KB407, that's Crystal Bio's HSV CFTR replacement therapy. Now, both of these clinical trials have recently reported protein expression data, showing expression of protein in cells of the lungs from people with CF. This is really exciting. And in the talk, we'll tell you a little bit about how to interpret that protein expression data. In the coming year, we expect 4D to complete their dose expansion study and provide a data update later in the year. And Crystal Bio is going to kick off their next phase, Coral 3, which is aimed to evaluate the safety and efficacy of their product in people with CF. So it's also important to note that the FDA requires significant clinical evidence linking clinical trial endpoints, like FEV1, to the way a person feels, functions, and survives, and that's their language. So how is it that genetic therapies delivered to a cell can impact the way a person with CF feels, functions, or survives? Hillary's going to take over at this point and walk you through that. But before we do that, if there are any questions, we're happy to answer a few before we move on. Okay.
Awesome. I think everyone understood everything. That's okay.
So I have two daughters with CF in their 30s. They have two copies of R1162X, so which is kind of bizarre. But my oldest daughter was in one of these trials, and I want to say it was in that VX522, because she was one of the ones that couldn't tolerate it. It literally, her lung function went boo, and so, which was scary. But I'm just wondering about managing those kind of things in these trials, because we were so proud of her for agreeing to do a trial, and then it's like, okay, your usual 88 lung function is now 60. This isn't good.
Yeah. Well, first, thank you. I'm grateful that she's participating. This is how research moves forward. It's nerve-wracking, but thank you. So we are seeing some inflammatory responses across the board, not only in clinical trials that are currently ongoing, but if we think back to Translate Bio, which was an earlier clinical trial using a lipid nanoparticle, and even as we now continue to study lipid nanoparticles in preclinical systems, we are realizing that lipids, as inhaled lipids, could have inflammatory responses. And so as we think toward the next generation of therapies, that's one of the things we're tweaking. And we're also trying to better understand how lipid nanoparticles interact with the immune system. And Hillary will touch on this later. Yeah.
Yeah, maybe I would just add 2 points. One is that, I think that goes back to Kara's comment around the Data Safety Monitoring Board, and that, they're really looking at, unfortunately after it happens in one individual, but really making sure that trials are proceeding in a safe way, and when they see these things, that's being carefully reevaluated. The other thing I would say is that there are some things that you really can only see in humans. And so as we think about the models, right, like if we think about cells in a dish, they don't have immune systems, and even as we think about animal models that are used, one context that I found helpful, maybe I'm not supposed to talk about animals, but I'm going to, but one context that I found helpful is if the human immune system is really very sensitive. And the reason for that is we've evolved over time to live in large groups, right? We have New York City, we have millions of people living together, and even over time. So our immune systems have evolved to be really sensitive. If you think about, other animals, whether that be mice or monkeys or other animals, none of them live in groups more than like a couple of hundred. And so as you think about, I find it helpful to think about the evolution behind those mechanisms. And so you can do all the testing in preclinical models. Nothing is going to mimic the human. Even the ones that are as close as possible, nothing is quite going to mimic the human immune system. And so unfortunately, there's some things we really can only learn in the context of humans. And the immune system is one of those things that's just different in humans fundamentally than in any other animal. I don't know if that's helpful if people like to think about evolution, but. All right. So, let me just get all right. So in this next section, we're going to switch gears a little bit, and we're really going to zoom in and focus on some of the cellular and molecular considerations for CF therapies. And data from our research community has really helped us to understand where in the lung dysfunction occurs in CF. And that information tells us where CFTR restoration needs to happen in order to improve lung function. So data suggests that an inhaled therapy would need to travel down the trachea and reach the conducting airway in order to improve lung function as measured by FEV1. So the lung airways are often described as an upside-down tree. So if you think about the lung and you turn it upside down, the trachea is the trunk, and you have lots of different branches of different sizes. In this analogy, what we really need to do is we need to restore CFTR in the small and medium-sized branches, in particular, of our lung tree. We don't think it's going to be enough to just restore it in the trachea or the trunk. It's not going to be enough just to restore it in the leaves, in the alveoli. We really need to be looking at those small to medium-sized branches, that conducting airway. And so if we zoom in a little bit more to the surface of one of those small or medium-sized lung branches, we can look at the surface of the airway, which is the lung epithelium. And whether or not we are delivering CFTR gene replacement therapies or gene editing therapies, we really want to target that same lung region, the small to medium branches. However, the cell types are different. The target cell type for a durable gene editing therapy is the basal cell, which is the stem cell of the lung epithelium, whereas the key target for CFTR replacement therapies are secretory cells, because these are the cells that naturally express a lot of CFTR. So we're going to talk a little bit more about that in the different cell types. So on the left is a schematic of the different cell types. And in the right is some real data from real experiments looking at the percent of CFTR expression in the airway epithelium. Now, ionocytes are cells that express a lot of CFTR. However, they're a very rare cell population. Maybe only about 1 in every 100 cells in the epithelium is an ionocyte. And so because they're so rare, but they express a lot of CFTR, in general, they express a moderate amount of CFTR. In contrast, ciliated cells, shown in blue or in that second part, really express little to no CFTR. The big bulk part of the graph here in yellow and orange and red is the secretory cells. And there's a couple of different colors because there's a couple different flavors of secretory cells. But in general, secretory cells as a whole are pretty common in the lung epithelium, and they express a good amount of CFTR. And so this is why they really are the key target for CFTR restoration therapies, and that's because in healthy individuals, secretory cells express the bulk of the CFTR, and we want to recapitulate what healthy looks like. Now, the basal cells on the far right really actually express little to no CFTR as well, if you look at the bottom two shades of green. The B to S indicates basal cells that are in the process of differentiating into secretory cells, and the B to I denotes cells that are in the process of differentiating into ionocytes. And so while these cells are not yet fully secretory cells or fully ionocytes, they're well on their way, whereas the true basal stem cells really express little to no CFTR. So we talked about ciliated cells and how ciliated cells really express little to no CFTR, but this also begs the question, well, what if they do? what if ciliated cells did express CFTR? And in the lab, we can actually force ciliated cells to express CFTR. Again, they don't normally express CFTR, but if we make them express CFTR, actually this does not result in CFTR function. And so I'm going to describe an analogy in the next few slides to explain why we think this happens. So to set up this analogy, bear with me a little, let's imagine the different cell types of the lung epithelium as vehicles. So if cells are vehicles, the secretory cell is a fire truck. Secretory cells naturally express CFTR, and expression of that CFTR results in CFTR function. Now, secretory cells move chloride through their CFTR into the airway. And so in this analogy, the CFTR is pumping chloride just as a fire hose pumps water. So the secretory cell is the fire truck, CFTR is the fire hose, and chloride is the water. So if secretory cells are fire trucks, ciliated cells are Toyota Corollas. No offense to Toyota Corollas. Ciliated cells do not naturally express CFTR. We also know that expression of CFTR in ciliated cells is not going to result in CFTR function. And that's because if you put CFTR onto a ciliated cell, if you put a fire hose onto a Toyota Corolla, what do you think is going to happen? I mean, it looks kind of weird, right? Not much, right? You can put a fire truck, you can put a fire hose on a Toyota Corolla, but like it's not going to result in water being pumped out. It just doesn't work that way. So if we think about, really the biological basis of some of my corny analogies, that's the reason that a secretory cell is a fire truck is a couple. The first is that it's not just about CFTR. It's not just about having that fire hose. You need lots of other parts in order to make that fire hose work. The fire truck also has a water tank. It has valves. It has lots of other things that are all important for making sure that that fire hose can work. And I think the most intuitive thing is that in order to pump chloride out of a cell, you need to have enough chloride. And so in order to get enough chloride into the cell, you need to have other channels that are moving chloride into the cell so that CFTR can do its job and move chloride from the inside of the cell into the airway. The other thing that is probably a little less intuitive is that if you're just pumping negatively charged chloride, bicarbonate is also negatively charged, out of the cell's going to get more positively charged. And that's not necessarily a good thing because cells need to have a pretty narrow window of charge. And so in secretory cells and fire trucks, there's mechanisms in order to pump positively charged ions out of the bottom of the cell in order to balance the charge change that occurs with chloride leaving the cell. So there's lots of other things that are involved in making a secretory cell be able to pump chloride beyond just CFTR. So if we summarize that a little bit and move away from my dorky fire trucks, if we summarize, data to date suggests that delivery of CFTR to epithelial cells that don't normally express it, like ciliated cells, it's likely not a problem. We think it's pretty net neutral, but it's also not going to be a solution for CF. Just like putting a fire hose onto a Toyota Corolla is not going to fight a fire, putting CFTR onto a ciliated cell is not going to cure CF. It's also important to deliver enough CFTR to the fire truck cells, for example, secretory cells, that have the right cellular machinery in order to enable chloride transport to CFTR. Now, we've talked a lot about epithelial cells, and they're a really critical part of the lung, but there's also a lot of other cell types in the lung as well, including immune cells. Immune cells can also take up CFTR gene replacement therapies, and this also will not restore CFTR chloride transport or provide therapeutic benefit. We talked about ciliated cells as kind of being this net neutral for gene replacement therapies. They're not going to, have a positive benefit, but we don't think they are really doing any harm. With immune cells, the jury's really still out. Immune cells can detect genetic therapy delivery vehicles and other things which could contribute to the activation of the immune system. And the impact of this type of interaction is really an active area of research and something that we're still looking to better understand. So if we put it all together, what we really need is we need to be targeting a genetic therapy to the right part of the lung, to the small and medium-sized branches. We need to actually get the delivery vehicle there, and we need to look at the cell types. So those are really the key pieces. And in this next section, I'm going to talk a little bit about how we do that in the context of looking in a human or in the context of a clinical trial. And the first step really is an airway biopsy. And an airway biopsy is important to really look in the right place in the lung, go and sample some things from those small and medium-sized branches of the lung in order to look in the right place and see what is happening there. The second thing that needs to happen is you need to actually look for the genetic therapy. And there's a couple of different ways you can do it. This is data that was kindly shared with permission from Crystal Bio. And in this image, on the left-hand side, they're showing an H&E stain, which is a very general stain, kind of helping us to visualize what the tissue looks like. In the second panel is DAPI, which is blue, and this stains DNA, so helps us visualize where the nucleus of the cell is. And in the third panel, they're looking at a viral marker, which basically is saying they're looking at a part of the genetic therapy and seeing where the genetic therapy went. And in the far right panel is the merge of panels 2 and 3, so you're seeing both the red and the blue. And so what's really exciting about this is we can see that they are delivering a genetic therapy to a good number of cells in the right parts of the lung. So that's what we know so far. The really other critical aspect, again, is that cell type determination. We know we want to be getting to the fire trucks and not the Toyota Corollas. This image shows data from the CFF's lab out in Lexington from Jed Mahoney and Victoria Momeyer, looking at CFTR expression in ionocytes as well as secretory cells and club cells, which are a type or a flavor of secretory cells. And so it is possible to do this. The thing I would just note is that both the visualization of the genetic therapy as well as the visualization and cell type determination are technically very challenging. So if we go back to our data from Crystal Bio, they were looking at 4 patients with CF with various CFTR variants that are shown here and baseline FEV1 functions. In the third row down, you can see the total number of biopsies suitable for analysis. So this means that they took 7, 5, or 6 different biopsies from the right places of the lung, that small to medium-sized airways, in order to look for where the genetic therapy was. In the next row down, it says protein marker assessed. And so they were either looking for CFTR directly for the gene replacement or for the viral marker. So that's really steps 1 and 2 that we talked about. And what's really exciting about this data is if you look in the bottom row, you can see that overall, they're seeing in the right part of the lung that they are delivering a genetic therapy to high 20s to low 40% of the total epithelial cells, which is really exciting. However, what we don't know yet is which cell types took up the therapy and express CFTR. So that is something we just don't know yet. And so as we think about these exciting numbers, 29.4%. No, I mean which cell, like secretory cells versus ciliated cells, are they delivering the genetic therapy both to fire trucks and Toyota Corollas, or just to Toyota Corollas, or just to fire trucks, or what's the distribution? That's just something we don't know yet. And that's also why it's really critical. Kara talked about FEV1, and there's a lot of pieces that go together in order to figure out if something worked. But if you just look at FEV1, that kind of sums up all of those pieces and tells you. So we try and look and get a sense of what we think it's going to look like. But really, FEV1 is really that key measure, and that's what the FDA is looking for. So as we think about kind of these overall exciting numbers, 29.4, 41, 42.1, that kind of also begs the question of, well, how much CFTR is enough? And so here we're going to define kind of normal CFTR expression, whatever that means, as 100%. So we know that if someone has 0% of normal CFTR function, then that individual has CF. We also know that healthy individuals have, let's assign them an arbitrary unit of 100% CFTR normal function. That person does not have CF. We also know that carriers, which should have about 50% CFTR expression, do not have CF. We also know that there's a decent amount of variability in CFTR expression in healthy individuals as well as those with CF. So if we were to take all of the non-CF individuals in this room or at VLC, our levels of CFTR expression would vary probably by about 5-fold or 500%. This is a decent amount of variability, and this is just biology. And this is not specific for CFTR. This is true of a lot of different proteins within the body, and that's what makes people unique and feel and function and look differently. But there is a decent amount of variability. And the reason I bring that up is because there's variability, we also think there's not like a cliff. It's not like a 50% carrier is okay, but if someone had 49% function, like that there was a sudden drop off and that person would have CF. So we think there's a decent amount of range there of what healthy can look like. At the same time, lab studies suggest that about 10 to 20% of CFTR expression can restore function. And this is pretty consistent with our understanding of about that 5-fold variability in the context of a CF carrier. So if we put that all together, really the takeaway here is about 10 to 20% of normal CFTR expression in target cells may be enough to ameliorate CF disease. So we've talked about some of the cellular mechanisms, but let's kind of dig into that a little bit more. So how does uptake by secretory cells, our fire truck cells, that results in about 10 to 20% at least of normal CFTR within the right region of the lung where CF dysfunction occurs, improve FEV1 in people with CF? So in CF, cilia function is reduced, and this is because the airway surface liquid, or ASL, is really small in people with CF, which means that kind of this mucus is kind of smushing the cilia. The cilia aren't able to beat, and the mucus isn't moving, and it's not clearing away pathogens in the same way that it should. When CFTR expression is restored in secretory cells and other naturally CFTR expressing cells, this induces chloride ion flow, and then water follows the chloride into the epithelial cell surface, leading to increased airway surface liquid, or ASL, and hydrated mucus. This allows the cilia to beat and improves mucus clearance. That clearance improves mucus plugs and improves airflow. And over time, that cycle of mucus retention, infection, and inflammation is decreased, which improves lung function. So I know I bashed on ciliated cells a little bit, but ciliated cells are actually really important. They're just not the target of CFTR expression. So the secretory cells are expressing CFTR, and the ciliated cells are moving the mucus. They just have different jobs. So in this analogy, in the fire truck analogy, you can think about maybe the Toyota Corolla is like bringing in additional firefighters, right? Maybe they're carrying extra firefighters. It still serves an important role. It's just not the role of expressing CFTR or having that fire hose. So we've talked a lot about what ideally will happen in a CF gene replacement strategy. But I think it's also important to think about, well, there are situations in which we could envision that we could see CFTR protein, but actually no change in FEV1 if there's not enough cells that receive the CFTR genetic therapy, if we're not hitting that kind of 10 to 20%. If too many of the wrong cells get the CFTR genetic therapy and then there isn't enough left over for the right cells, Kara's going to show a schematic of this in a couple slides that hopefully will help clarify that. It's also possible that the right cells get CFTR, but the immune system reacts and causes inflammation, which kind of offsets that positive change in FEV1, or just that CFTR needs to be restored longer in order to drive that chain reaction to improve FEV1. So with that, I'm going to pass it back to Kara to tell you about the next generation of CFTR gene replacement therapies.
Okay. Any questions before we move on? Okay. All right. Thanks, Hillary. So we know that current CFTR replacement strategies can successfully deliver to cells in the airway, but we don't know if enough of that drug is getting to the right cells. And although we're really excited to see the clinical trial results that come out over the next year or 18 months, we're not going to sit and wait, right? I told you that at the beginning. We're already evaluating risks, and we're looking toward version 2.0, which is really focused on targeted delivery or targeting specific cell types of the lung. And this is because, as Hillary, I think, delivered clearly, we need to target the fire truck cells, those secretory cells. This is where CFTR can function and benefit people with CF. So how does cell targeting work? Even though we show cartoons like this all the time, CFTR is not the only protein on the top of secretory cells. In fact, there are many proteins that decorate the surface of secretory cells, and in fact, all cells. There's many, many proteins. And in the lab, we have tools like antibodies or other molecules that can be used to detect these proteins because they have natural properties of really tight and specific binding. Now, antibodies and tools from the lab can actually become part of a therapy if we attach them to a delivery vehicle, like an LNP, and I show that on the left. And while on the left it looks simple and easy, just boom, there they are, all the little antibodies on the LNP, this is actually a whole body of research. There are many different ways to attach an antibody. There are many different directions an antibody can face, and it has to face the right one. We need to think about how many we want and how they affect the overall stability, charge, and properties of that delivery vehicle. All of that work is ongoing. But assuming we can do it successfully, on the right, we see a lipid nanoparticle with targeting antibodies binding very specifically to that triangle protein on the secretory cell. That's what we want. So how do we find the right protein? When we think about proteins on the surface of cells, there are some that are expressed equally on all cell types, or maybe equally on secretory and ciliated cells, like this purple protein. There are some that are specific to ciliated cells, like this orange rod. This is not what we want to target. This would take our CFTR to the wrong place. The red triangles are only on secretory cells. And ideally, this is the type of protein we would identify and exploit to target our genetic therapies to secretory cells. But biology is rarely ideal. And so it is unlikely that we will find a protein that is very highly and specifically expressed on secretory cells. There will probably be some of the orange rod on secretory cells and some of the red triangle on ciliated cells. And so what we're looking for is a protein that is mostly expressed on secretory cells versus ciliated cells. And in science, we call this enriched. We're looking for a protein that is enriched in our desired target. Now, unfortunately, we haven't found a protein that is enriched on secretory cells yet, but we are working very, very hard on this and have several new programs that are aimed at finding these types of proteins. So we've gone on and on about targeting. Why is it so important? And the answer is because we really think that this approach can improve both the efficacy and the safety of a genetic therapy. Let's go back to Hillary's fire trucks and fire hoses. We know in this example that we need 4 working fire hoses for our drug to be effective. We also know that we can't deliver more than 6. No more than 6 is safe. So if we look at an untargeted therapy and we deliver 6 fire hoses, they're going to distribute equally across the fire trucks and the Toyota Corollas. 6 were delivered, but only 3 fire hoses work because only 3 hit fire trucks. This therapy is not effective, and we can't go up in dose because only 6, right? Only 6 is safe. So let's look at a targeted therapy. Again, we're going to deliver 6 fire hoses, but this time they're actively looking for fire trucks. And so out of the 6 that were delivered, 5 of them hit their target, and we have a therapy that is both effective and safe. So why are we really excited about this? It's not because of fire trucks. It's because we've seen it works in other systems. We're going to get into real data now, and so bear with me. There's no more trucks and hoses here, but I'll try to explain it. We've seen targeted delivery work in the liver. In this example, we're looking at an antisense oligotherapy that is either untargeted in the panel on the left or the same version of that antisense oligotherapy that is targeted in the graph on the right. Now, this particular antisense oligo is different than what we're doing in CF. It's designed to reduce expression of its target. So for the context of this example, when those bars go down and get smaller on the graph, that's success, okay? That's what we want to see. So this therapy improves, targeted delivery improves this therapy in 3 specific ways. The first is by increasing overall effect. If we look at the left, we find the lowest gray bar, and we draw a line across, and we see the value is 40. If we look at the targeted therapy on the right, we do the same thing. We find the lowest bar, draw the line across, and we see it's all the way down to 20. So this signifies a greater overall effect achieved with the targeted ASO. Second, we can look at dose. On the left, the greatest effect, the smallest gray bar there, is seen at a dose of 24 milligrams per kilogram. When we look at the dose that achieved the greatest overall effect in the targeted therapy, it's only 10 milligrams per kilogram, more than half of the untargeted. So we've seen increased overall effect at a lower dose. Finally, we can see from this graph that the targeted therapy is more specific. If we look on the left, we see a decrease as dose goes up in both the gray bar, which is the cells with our target, and the black bar, which is cells that don't have our target. So they shouldn't change. When the black bar goes down, that means we're seeing non-specific effects. If you go to the right, those black bars are tall. We're not getting non-specific results. We're only seeing decrease in the gray bars, which is what we want. And so we've seen our effect is not only greater at a lower dose, but it's specific to the cells we want to target. Right? It's pretty powerful. Question? This is not at all related to CF. This is just a good example of what we've seen in the liver and why we're so excited. So we do hope we can do the same thing in CF. One possibility is that our current therapies will hit some secretory cells. But if too much of that therapy goes to ciliated cells or immune cells, the therapy may not be effective enough to fully restore lung function to a level we can detect in a clinical trial using FEV1. But if we can find the right target to bring our therapy directly to where it needs to go, to those secretory cells, we can get more CFTR with less drug. And we hope that this will improve not only the efficacy, but the safety, and that we can see the results that have been generated in other diseases and therapies that target the liver. Now, we focused most of this talk on non-permanent DNA and RNA therapies to restore CFTR. And we've talked about why delivery vehicles are so important, why cellular targets are so important for creating a therapy that is both safe and effective. But it's important to mention that these same properties are also really important for genetic therapies like gene editing. We believe that many of the learnings from RNA and gene therapy are going to help us progress faster as we apply them to gene editing programs that we are working on currently. And although there are no gene editing therapies in the clinic right now, we continue to work toward this and believe that gene editing is the best hope for a permanent transformative therapy for CF. So in conclusion, we expect to see updates on CFTR replacement trials in the next 12 to 18 months. And while we're looking for early signs of efficacy, we know these can be complicated to interpret because they're on the context of real people in real life, and they're very small trials. We know that in order to be effective, the therapy must deliver enough CFTR to the right cells. And this is challenging to evaluate directly, especially in people. And so we really need these therapies to show improvement in lung function. This is what the FDA is looking for, and this is going to be critical for evaluating our clinical trials. While we wait for these results, hopefully, we're going to work on the next generation. Targeted delivery is a focused area of research that could improve genetic technologies, and efforts to apply this to CF are early, but we are learning from other indications like the liver. And finally, although gene replacement was really the focus of today's talk, we continue to make really rapid progress in gene editing. It's really exciting, and we hope to update you on that soon. Until it's done, thank you very much.
Thanks, Hillary. I just had a question on that slide that you showed where it was 0% CFTR function to 100, and you were at the 10 to 20%. Where is modulators taking us? Like, is it, if we say like the modulator that we have right now, the Trikafta, like they said yesterday that we were looking at like 100%. What does that percent look like compared to what you're saying, 10 to 20?
That's a great question. Hillary, you want to go?
Yeah, I think one thing I would say is that we know that people respond differently to modulators. And so that's a very difficult question to answer in the context of like overall. We know that some people respond really effectively to modulators and have really big increases in CFTR function and, you know, are probably pushing up there in the green, whereas other people don't respond particularly effectively. So there's definitely a range there. But yeah, I think, anything you want to add there, Kara? It's really hard to put in, it's a great question. It's really hard to put a number on that.
How are they saying that Trikafta is at this percent, but their next drug is at 100%?
So one thing that people do in various studies is in cells in the lab, they will take lung epithelial cells from like healthy individuals and compare those. And so they can say, okay, you know, this modulator resulted in this much CFTR function. We can actually measure that. We can measure how much chloride is moving. And we know that a healthy individual has, you know, this amount of CFTR function. And so then you look at the percentage. The challenge with doing that is I talked about variability, right? And so just like, you know, carriers have variability, healthy individuals have variability. So where you define that percentage that you're then normalizing that to makes a big difference. And so we think about, like, if you look at cells from, you know, person one versus person two versus person three, that number is going to be different. And so I actually don't love when people are like, oh, this is like X% of wild type. I'm like, well, what's your wild type? And so how you define that is difficult. But that is where those numbers are coming from, is a lot of it is like cells in a lab. But I think it's tricky because you're normalizing it to something, and what you're normalizing it to can vary.
Yeah, it's a range. I was going to say the same thing. And one thing we do look at when we look at genetic therapies in the lab is we compare them to Trikafta. And so we say, how close are these to Trikafta in a dish, because we know that if they get close to that range, that they should be effective.
Yeah, and so in terms of like those studies in the lab, as Kara said, I actually think it's much better if you have, for example, cells that have like one copy of F508del and one copy of, let's say, R1162X, and you can say, okay, this is what percent of that genetic therapy as compared to Trikafta in this like given individual, and that tends to be a little bit more meaningful just because of that variability.
Hi, I was curious whether finding the right target is what the difficulty is, or finding the right target with a delivery mechanism that would work for that. And then if it is finding the right target, what makes that so difficult?
Yeah, so it's both. When we think about drug development, lipid nanoparticles seem easy to make, right? You throw some lipids in a tube and you shake it up and there it is. But this has to be turned into a drug. So you have to be able to make it reproducibly. You have to put it through a nebulizer and hope it doesn't fall apart, and hope you can dose enough of it at a safe range. So developing the delivery target or delivery vehicle on its own to be a medicine is really hard. Finding the target is difficult because we have to compare secretory cells to all the other cells. And as Hillary talked about, there's a lot of variation in biology. Biology is not ideal. And so you have to look at a lot of different cells to get conclusive results. And so finding a target that we think is right is part of it. There's also a lot of science I didn't go into. So how long that target stays on the membrane. So something that naturally happens is membrane-bound proteins recycle. That protein gets old and it gets pulled back in and degraded and it comes back up. And so the rate of recycling, there's about 1 million biological factors that make identifying the right protein hard. And then the third piece is putting them together. There's actually a lot of different ways to put something onto a lipid nanoparticle or a viral-like particle or any different particle, and making sure it's facing the right way, it's spaced correctly, and it doesn't affect the druggable properties of that delivery vehicle are really hard. So all of the above.
The presentation we saw yesterday, I'm over here, they listed lipid nanoparticles as one delivery mechanism, but also polymers. And I haven't heard anything since that presentation. Can you elaborate a little bit on that as a delivery vehicle?
Yeah, you want to go, Hillary?
Yeah, happy to. So lipid nanoparticles, really, their development as a delivery therapy, as a delivery vehicle for genetic medicines was really accelerated during COVID. And so lipid nanoparticles were used as part of COVID vaccines. And so that research got a ton of focus and was really accelerated. We are very excited about polymers. There are some specific features of them that we think make them differentiated from LNPs and may be very compelling. We are actively looking at companies. I mean, I can think of a couple right now, but we're very actively looking at in terms of their polymer technologies. So we continue to pursue both LNPs as well as what they're called PNPs, polymeric nanoparticles. It's just in general as the field, PNPs are a little bit less further along, but that is something we are very excited about.
Yeah. So lipids are a type of a polymer. And so when we think about polymer nanoparticles, the space is huge. There are way more types of polymers we could use than types of lipid nanoparticles. And so not only is it a newer field, but it's a much broader field. And so we're really, Hillary's team is really sorting through all the technologies to try to find something that we think will work. The good news is there's some exciting things that make them look better. Like maybe they're easier to make and maybe they're more stable and they could have potentially less inflammatory side effects. So we are actively pursuing that space. It's just early. Go ahead.
I know there's a lot of interest in gene therapies, especially for the people who are not eligible for modulators. I guess I'm curious, how does this research, is it different depending on different people's mutations, or is it mutation agnostic? Is it like, do the trials run only on specific variants, or how do you think about the different genes?
Right, it's a great question. So all of the different mutations that cause CF are all in the same gene. They're all in the CFTR gene. And so a therapy that gives a new copy of that CFTR gene essentially has all the correct bases. So that would work for any mutation, right? It's a whole new copy and every single nucleotide in that is correct. So it doesn't matter what mutation you have, a replacement therapy that gives a new copy of mRNA or a new copy of DNA will work. Some of the technologies we talked about, like gene editing, where it goes in and corrects a certain mutation, are designed to be mutation specific. Now, as those technologies advance, we can start to do tricky things. We can design one editor that could potentially correct any of a few mutations that occur close together. So if somebody had G551D and somebody had R553X, there could be one editor that could work for either. But generally speaking, if we think about a technology that gives a whole new copy, that'll work for anybody. And editors and ASOs are specific to mutations. She's coming.
Come to you.
There are people online, so they asked us to use them.
Yeah, we can hear you, but.
So you gave an example of a small participant. You had mentioned like 3. I was just curious, is there kind of a minimum participation level that would make the study accelerate? Because with 3, maybe like you had mentioned, somebody got sick and then there's not as data. And is there kind of like a minimum threshold? Obviously more is better, but you may not get 1,000 participants. But
Right. So we're never going to get 1,000 participants again, right? Long gone are the days of modulators because we're really targeting with these genetic therapies, people who are not eligible for modulators. Although, like you saw Crystal's data had people with F508, they are accepting people who are on modulators into many of these trials. The hope is that they'll first be available for people who are not eligible. And so that's a small population. And so as we think about clinical trial design, we want to enroll enough people, but we also want to move slowly and we want to be cautious. And so the general approach is to start small, so each dose is just a few people. And again, we're looking for safety, not efficacy in these small numbers, really. And so even if there's an adverse event, which we hope we don't see, but if we do, is it safe enough to keep going? Then we go up a dose, and it's 3 more people, and it's 3 more people. And so as we go on and we get a better sense of safety, then we build out those numbers. And when we think about statistical significance, one of the challenges of these genetic therapies is that in these small numbers, which we're going to have because our population who is interested and motivated and participating is small, we're going to need to see a big change in FEV1 to see statistical significance. The good news is that the regulatory landscape of the FDA is currently evolving, and they are developing new pathways and new ways of looking at data in clinical trials for rare diseases that are a little bit more flexible. And so we want to take advantage of those, but at the same time, be really mindful of safety and protecting our community. So I don't have a great answer other than it's a balance and it's a constant conversation.
Maybe I'll just add like one small comment to elaborate on that. And really that's like, we all want to see that, like that sign of efficacy, right? But again, those phase 1 and phase 2 studies are really designed to look at safety. And so they're not going to have those number of participants. When you start to move into phase 3, also known as pivotal trials, there's actually something that's done called a power analysis. And so that means if we look at how effective we think the therapy is going to be, you can actually calculate how many people you need for that clinical trial, you estimated, in order to show a statistically significant difference. So there's like actual calculations that are done to what we call power those statistics, but those are done for the pivotal, the phase 3, in order for efficacy.
And if you went to JP's talk, he may have talked about a study called REACH, which is looking to really characterize our population of people with CF who are not eligible for modulators. And the hope is to be able to use this group as what we call an exogenous or an external control group. And so one strategy for a small clinical trial is to have very few placebos or even no placebo, and to use external controls to enable that statistical analysis. And so that allows a trial to give the drug to all the people in the trial and then to evaluate against natural history. And the FDA is becoming more comfortable with these types of strategies. So this is something we're looking toward in the future.
So once we've mastered a specific genetic makeup, how rapidly will it be to move to the other ones through the ASO capability?
So for ASO or for gene editing, sorry, this sometimes is really loud. I'm going to talk about gene editing instead of ASOs because Steve Rowe talked yesterday pretty excitedly about baby KJ and his story. And so this story has really been a catalyst for some of the regulatory changes I'm talking about. And so what has changed is the FDA has realized that for a genetic platform technology, the things that change between an editor that targets R553X and an editor that targets G542X is not very much at all. And if they use the same lipid nanoparticle and the same gene editor, and the only thing that changes is the targeting RNA, that those therapies aren't that different. And so they have decreased the number of additional studies that have to be done for each additional mutation. And so for a while, we talked a lot about super exon, which we're still working on, because we didn't think mutation by mutation was possible. But in this new regulatory landscape, the flexibility that has been opened up has made us realize that actually we could go mutation by mutation. And so each one that you would add on, we believe will have a much smaller data package associated with it because we already know so much from the first one. And the changes are very, very minute. So hopefully faster. And those are the things we're thinking of in the editing programs we're working on. And that's why you saw Steve like right on the edge of his seat, pretty excited yesterday.
Yeah, I think that's great context for the editing therapies. The only thing I would add there is just in general, you know, you've heard it from us, you've heard it from Steve, you've heard it from others. Delivery is the biggest challenge right now for genetic therapies. And so if we can solve the delivery problem, iterating through additional, mutations or additional genetic therapies, we think will be much faster than getting that like delivery vehicle that's really effective.
Good point.
And so kind of similar to that, I know that the urgency is really around the lungs, but thinking, jumping ahead to the future and thinking about a whole body cure, do you think it's going to be organ by organ that's affected? Do you think there's going to be a way to deliver whole body? Or what do you, I know it's a crystal ball probably, but what do you see?
Yeah, my crystal ball isn't that good, I'll just say. And the conversations that we're having today around gene editing are so different than the conversations that we were having 10 years ago. And so I think that it's just, and things are evolving really rapidly. And it's a great question. I think it's possible that you could have a systemic therapy that would go to the right places. I think, organ by organ, or maybe, other sorts of routes are also still on the table. But yeah, sorry, my crystal ball's not that good.
My crystal ball would say with a lipid nanoparticle, you'd have to go organ by organ. But as we see evolution in delivery technology, that may change.
There's a lot of talk about urgency and acceleration. How are we using AI at all to facilitate some of this research?
Yeah. So I think in our area where we see AI the most is when we talk about this targeted delivery and identifying target proteins. So I mentioned that putting a protein on a lipid nanoparticle or on the surface of something is actually really hard. And so there are a lot of companies using AI to design protein binders. The reason we see a lot of AI here is that AI is best where it has a lot of data to learn from and it knows the rules. And so we've learned over the many decades of research how proteins work, how protein binding works. And so there's a really robust set of data we can use to design novel protein binders. And so that's where we're seeing it the most. I don't know, Hillary, you want to add?
I think that's really the heart of it.
Yeah.
So the other thing I would say is I know we're like 10 minutes over time, and so I want to be respectful of your time and let some of you go who would like to go, but Kara and I are happy to stay for a few minutes and address any other questions.
Thank you so much.
Thank you, guys.